Welcome to the NeuroExpress tutorial video. NeuroExpress is a Windows program designed for the analysis of various types of electrophysiological data acquired in patch clamp experiments. In the first part of this video series, I described the installation of the program and how to analyze voltage responses of neurons receiving current step stimulation. Today I will mostly talk about the analysis of minis and spontaneous postsynaptic currents as they are recorded in voltage clamp experiments, typically in wholesale patch clamp configuration. Synaptic current traces are recorded in gap-free mode of acquisition, and the sampling rate can be 10 or 20 kHz, but the program can handle data with 50 kHz sampling rate as well. These data files usually contain one or two channels. Of course, the membrane channel must be recorded, so that's the first channel, but the second channel in these files can be the holding potential. In this example, the program detected the holding potential channel, and that is shown here. It was minus 71.4 millivolt for this recording. And of course, the membrane current or the synaptic current events are shown in the top and middle panels. So the program loaded the file, analyzed the structure, found the current and voltage channels in this gap-free recording, and performed the analysis pretty much instantaneously after loading the data. So let's get familiar with the program desktop and see how these different panels represent our data and how we show the results of the analysis on these plots. So in the middle of the desktop we can see the full trace, but actually there are these buttons with the plus and minus sign that we can use to decrease the time scale or time window that is shown here. Also, the scroll bar can be moved across the entire recording like here, and then we can look into smaller details of the recording. In addition to this scroll feature, we have a zoom window. We can define a little zoom rectangle by clicking with the left mouse button like here and draw this yellow colored rectangle. So this is the zoom window or zoom rectangle that we can use to look into the recording and see even smaller details of the recording. By clicking in the middle of this little rectangle, we can drag it across the recording like here. If you click outside, then it will just disappear and it will unzoom. Then you can go back here, or you can also click on the full button to show the entire recording like here. So this is basically how we navigate through our recording, and we can see very tiny details or the entire recording as we like. So the display is fast, and also only the selected part will be shown on the top panel. So the title here says selection from full trace, and in this case the selection is within the zoom window, but if I unzoom, then the middle and the top panels will show exactly the same current samples. On the bottom part of the desktop we can find four plots that will show the results of the calculations and the results of the event detection that was performed previously. So here in this example, the inter-event intervals are shown as a function of the elapsed time, but you can change the content of each plot by bringing up the pop-up menu, by clicking on the function button here, or just by right mouse button click. And then you can select the feature or the parameter you would like to see. For example, I would like to see what was the rise time of the minis, and this plot will show the rise time as a function of the elapsed time. Also, the time constant of the events are shown here, and you can access all these different parameters and different plots by the right mouse button click. So another important feature is that when you zoom into your data, then the content of the zoom window will be shown as a current trace on the top, and the associated parameters like inter-event intervals and mini amplitudes will be shown also, and these plots will be continuously updated. So the time scale on the top panel in the zoomed current trace will be the same as the time scale applied for these calculated parameters. So you can scroll through the entire recording and see how the event amplitude or inter-event intervals changed during your recording. Also, you can see that the program will calculate the median standard deviation of these calculated parameters. It will also show some numbers like 6,487 events are shown within this plot. Of course, if you zoom into a narrower section, then the number of events will be much smaller. The average or mean event rate is also updated here. In this example, the event rate was approximately 5.5 Hertz. But if you go through, then you can see that this number is continuously updated. In the demonstrated example here, the baseline of the recording is 0, and that is caused by the high-pass filter that we used during the acquisition of this data. But in many cases, we are not using high-pass filter. We only use the low-pass filter to smooth the data a little bit, and then we will see that the holding current can drift a little bit. In the next example, let me show that this can indeed happen. So the baseline current, the holding current can go, and in most recordings we will see a little bit of fluctuation. Still, the program has no problem finding the events and finding the minis, even when the baseline is moving up and down a little bit. So let's talk about the event detection algorithms in the following. Basically, we can choose between two different algorithms, and I mostly use the first one, the local minimum and maximum analysis. This is a very quick and very reliable algorithm, although it has some drawbacks or limitations, of course. So we can optimize, or we can certainly help the algorithm to find the events the most accurately and most reliably by editing these three parameters and maybe applying the smoothing in the fourth edit box here. So basically the first three parameters are the ones that we need to adjust for the recording in order to get the most reliable and most accurate event detection. The first parameter here is called the estimated noise level, and this is the parameter that we want to set the most carefully, because this will greatly affect the accuracy and reliability of the event detection. So how to do that? The best method is basically to zoom into our recording and read the peak-to-peak fluctuations. So in this example, we can see that the holding current was fluctuating between -70 and -90 picoamp. So a -20 would be a good choice for the event detection. So when we set this parameter to a higher value, then the total number of detected minis will decrease. When we decrease the noise level, then the number of events can increase a little bit. We can also adjust these parameters by clicking on the edit box and use the mouse wheel. I can increase this parameter by rolling the mouse wheel up, or maybe down when I want to decrease. So this is a quick way to perform the analysis and to adjust parameters with the mouse, and the calculations will be performed immediately. So you can actually see how quickly these calculations are performed in NeuroExpress. There is also this feature auto estimate. So basically the program can perform the estimation of the baseline automatically. In this example, the program detected or determined that the fluctuations were approximately 21 picoamps. So that was our initial choice, and it was very close to this one. The next two parameters are used to describe the kinetics of the events to be detected. So these parameters can be set empirically, and you can also use the mouse wheel to play with these parameters. For example, the rise time parameter here can be upregulated or decreased by turning the mouse wheel. When you do that, the detected events will be updated in the EPSC panel on the right upper corner of this desktop. And then what you would like to see is basically that you have nicely grown, nicely peaking events, but their baseline sections are also very, very clean, and you have a stable baseline for most of these events. The next parameter is the decay time, and basically when you increase that, then the program will use a longer section of the current race to determine the decaying of the events. And basically you would like to see something like here, that all the termination points, these blue colored symbols, are within the plot, but maybe they should be a little bit somewhere at the end of this panel, like here. So this will show that most of these events are fully grown and decayed before the end of this panel. And of course it's a good idea to look into your data and just go across the recording and see whether these events will fit into this little plot. And when you see that, then you can determine that these rise time and decay time parameters are set correctly, and then your calculations will be as good as possible. There is also a smoothing parameter that you can adjust here. Smoothing can be performed in two ways. Either it is a simple moving average filtering or a Savitsky-Golay filter that you can apply. So increasing this number will perform stronger and stronger smoothing of your data. 0 means that the smoothing is not applied, but the raw data are used from the file. So this is like a post hoc filtering that you can perform to see the events more clearly. If you increase, then you can see that the events become very, very smooth here, and the fast little fluctuations will disappear. This can sometimes help the event detection, but you want to be a little bit careful how you apply this. Data that are already low pass filtered during the acquisition or during the experiment should not be filtered too much. Maybe you can use a smoothing parameter for 1 or 2. And then you can also use the Savitsky-Golay filter, which is a little bit better than the moving average. You can increase this parameter, and then that will not introduce so strong changes in the shape and amplitude of the events. So Savitsky-Golay filter is probably more appropriate for this kind of electrophysiological data than the moving average. But it's your choice. If your data are contaminated with high frequency noise, then you might want to use the smoothing here. There are a few other parameters here that are used to construct histograms from your data. For example, here the maximum inter-event interval is set to 1 second. So inter-event intervals from 0 to 1 second will be used to construct this distribution. You can increase this number by turning the mouse wheel. Now it's set to 2 seconds, or you can go back. And then the next parameter is the bin size. So that will basically determine the resolution of your histogram. If you increase this number, then the resolution is decreased, or you decrease this number, and then you will get more bins within the scope of the histogram. You can do the same for the amplitude histogram. And for both histograms, you can use the Parson method and the Parson estimation for calculating very smooth distributions of these probability data. Okay, so when you see the current trace on the top panel, you can see that some of these events are colored with the magenta color. Let's zoom into these data to figure out what is going on here. So basically the program will perform an automatic fitting of the data using a mono-exponential function, or you can use bi-exponential fitting also. But you don't need to adjust anything, because the program will certainly try to fit these events based on the settings that you have for the kinetics. So here, when we zoom into a single event, then you can see that the mono-exponential function is drawn here. And the amplitude, or the peak of the event, is indicated by this reddish symbol. The baseline, or the reference point of the event, is shown with this aqua color, and the termination point is the blue one. And the exponential here is calculated automatically. So you can see that most events that are detected will be fitted, and then the program will calculate these decay time constants automatically. So this is true for most events. And then if you display the mono-exponential decay tau, or time constant, here, then you can see that the majority of these events were fitted successfully. So fitted is 95.1%, and the total number of events was 1232, and 95% of these events were fitted successfully using the mono-exponential function. So then you will have these symbols, these data points describing the decay time constant as a function of the elapsed time. So again, you do not need to do anything to perform this, because the program will do these calculations automatically. Let's see another recording to demonstrate additional features of the NeuroExpress program. This is an axon binary format file created with the P-clamp suite and the P-clamp electrophysiological program. So here we can see the event nicely detected, and also the baseline. But there are also these vertical lines all over the recording. So what are these? If we zoom into the data using the zoom window, then we can see that there are these sudden big jumps in the holding current, in the voltage clamp recording. So what are these? Basically here the excess resistance of the electrode was monitored or checked using rectangular voltage pulses applied to the membrane of the neuron. These are of course artifacts, and we do not want to include them in the data analysis. These are actually contaminations that will cause some spurious data and wrongly detected minis. So when this happens, then the program will try to eliminate these artifacts automatically, and they will be ignored. Basically, the program will use the voltage channel in the recording, in the data file, to find these potential artifacts. So when the holding potential changes dramatically during the stimulation, and then the program will try to find these steps and remove the corresponding data from the current channel. Prior to the artifact removal, the trace looks like this. So this is actually the original current recording, and the program can perform the artifact removal also when we bring up this pop-up menu. So we can instruct the program to try to exclude these artifacts. So here we can see these big jumps in the holding current, and these were caused by the voltage stimulation that was applied to measure the excess resistance of the electrode. So we can use this attempt to exclude artifact menu to perform this removal of the artifacts, and now it reports that 12 such artifacts were detected and removed from the current trace, and we are back to the original nicely cleaned trace. If we want to exclude sections of the current trace manually, we can also do that. First, we need to bring up the cursors. So there is a blue and a red cursor to define a section of the current recording that needs to be eliminated or ignored. So when we set these cursors to the right place, right locations, then we can instruct the program to ignore this selected range. And now here this section will turn into gray, and there will be no events detected here, and no inter-event intervals or amplitudes will be calculated for these data points. We can also define sections in different locations, and we can ignore many more parts that are perhaps contaminated, or there was some sort of strange drift in the recording. So basically we can clean up the data using this feature, these red and blue cursors to set the beginning and the end of the sections to be ignored. Okay, so far I demonstrated the analysis of excitatory postsynaptic currents, but the program can work with outward currents or IPSCs like I show here. We need to check this checkbox, outward minis. So the program will look for positive changes in the holding current, and it will look for sudden jumps in the current. So positive direction changes. And then the analysis will be very similar to the analysis of the excitatory events. And we can see here that there are some slowly decaying events here, and unfortunately there are multiple overlapping events. So in this example, the analysis is a little bit more difficult because the overlap of the events will make the calculations a little bit more difficult. Still, the program will find the decay time constant and the rise time and additional parameters like area or normalized area of the events. One more thing that is useful to show is that you can change the scale of your plots. You can shift the entire scale, or you can just magnify the scale using this drag and click maneuvers like here. Okay, let's see a few other additional features of the program. In the options menu, you can see this one, show cell images when available. I use this only when I take pictures of the cells during the acquisition, and then the program can actually display these microscopic images. But when we do not have these JPEG files, then I can leave this menu unchecked. And then this little panel will show up, and then here you can specify some metadata associated with your recording. For example, you can specify from where you recorded your data. For example, here in this example, I can specify this was a hippocampal culture, And I can also specify what kind of neuron I was recorded from and what is the treatment group associated with this recording. Also, there is this little evaluation box here where the program will report a few findings. For example, how many events were detected, whether the baseline was stable or drifting, and additional information. So this is actually useful for a quick evaluation of the data, and the program will report these things automatically. Also, when you try to select some important sections of the data, the current trace, you can use the right mouse button click and bring up this pop-up menu here. So first you want to bring up the cursors, and let's do that. Show the cursors. So we have the left cursor, which is blue, and the red one, which you can move here. So when this happens, you can define a range of interest. For example, you decided to perfuse your preparation with a neuromodulator that can change how these synaptic episodes, synaptic transients are generated, and how many minis are basically created or produced during the drug application. So in this case, there is a control section, and then you can define a range of interest which is associated with the drug application. Again, you bring up the pop-up and you define range of interest. So in this case, you can say this is where you apply the drug, and then you will see that there is this empty control section and this highlighted section which indicates that here the drug was introduced. And of course, if you zoom into your data, then also it will show in the bottom panels where the drug is introduced. So now we can perform the analysis separately for the control section and for the section with the drug applied. Next, I defined a control section, so that is highlighted with this blue color here. So basically now the program will be able to calculate event parameters, amplitudes, time constant, and other parameters for the control section versus the drug application section separately, and we can do the statistical comparison easier. So when I have these data and we are satisfied with the program, how it detects the events, and we believe that this is already the optimal analysis, then we can send out the data into a third-party program. In this case, I will use an Excel worksheet to display the data, and this is where I want to export my calculated parameters. So I click on the send button here, and this is what happens now. Basically, the program filled up these rows in the Excel worksheet, and we have some metadata here that we already filled out for the recording. Of course, the first column will show the name of the file, holding potential, and some parameters that we set for the analysis. For example, the noise level, the algorithm that we used for the calculations, etc. The selection will be shown here. So we have basically two sections and the full trace. So the statistics for the full trace is shown in the first row. Here the selection starts at 0 seconds and ends at 200 seconds. The control is here, and it will start at 0 seconds and it ends at 120.9 seconds. And there is the third section, the drug application. And what can we see here? How many events were detected for the different selections, different sections of the current trace, the mean inter-event interval, the median of that, the standard deviation here. Also, what was the mean rate of events, which is in this column, and there are all these other calculated parameters, time constants, area, medians, and standard deviations will be shown. In addition to this this mode of using the program, we can also use the zoom rectangle. So we can define a temporary range of interest which we want to analyze separately. For example, I like this section of the data very much, and I would like to get the inter-event interval statistics for this particular window alone. So now I click on the send, and then the selection will turn into custom. So this row will contain the inter-event interval data, the amplitudes, etc. for the custom section. And this section starts at 9.47 seconds and ends at 69 seconds. And these are the calculated parameters for that range. But I can move this window and send out these calculated parameters for the different windows. So I can do this as many times as I want. So this is also very helpful feature of the program. Sometimes you do not need to define a range of interest. You can actually clear those, but you can still find the best parts of your recording that you would like to focus on during your analysis. We also have the option to export the parameters of each event detected by the program. So when we click on the list button here, then the program will fill up this table, and this table contains basically all the detected events, the number, the baseline or the reference current level before the event arrived, and the amplitude or the peak of the current, and when the peak was detected, what was the end of the event, so what current was used to determine the termination of the event, what was the inter-event interval associated with that particular event, their amplitude and area is also shown. So you can export your entire event data set into an Excel worksheet by clicking on the list here. In general, the program does a pretty good job in finding, detecting the events and calculating their various parameters like amplitude and time constant. However, you might be interested in finding these events manually or visually, and also to determine exactly where they peak or how they decay. So you can do that manually using the mouse. And to do that, first you need to click on the auto button here under the file type here. Here you will see that the label turns to core, which indicates manual correction mode. And on the top panel you will see a single a single synaptic event, and here you will have the option to move the baseline, the peak and the termination point individually. So let's do that. For example, I would like to move the baseline point a little bit before or lower. So I can move this with the mouse, and also I can change the location of the termination. So let's say this is good. Maybe the amplitude will be also changed a little bit. You can see that while I am moving these points, the time constant curve or this exponential decay is also updated by the program. So that can help the program to fit this mono exponential function the most appropriate way. Next, I can go to the following event that the program detected. I click here. Okay, this maybe I will move this a little bit forward. The baseline can be like here, termination a little bit back. So I can do this thing for each event. This looks a little bit ugly. Maybe the time constant is a little bit better now. I can do this for individual events. So basically, when I do all these manual corrections, then the program will remember how I located or positioned these current level points and their times, and these will be used for the calculation of the inter-event intervals, amplitudes, and other parameters. If you want to delete an event, then you can bring up this little pop-up menu. So here I will click on this event, and I can just click on delete, and that event will be not used for the calculations. If I want to insert a new event, for example, I want to put it back, insert a new event. Okay, maybe I just play a little bit with the points that define the event, and okay, now this looks more or less fine, and we are done. I can move to the next one. I don't want to keep this, so maybe I'll just delete. So you can do this thing manually for most events, but sometimes, of course, you can run into trouble when there is such a huge overlap. In this case, you might want to move this termination point a little bit earlier, like here, and maybe you want to insert an event here and do something like that. It's not absolutely proof, not absolutely safe method, but you can definitely clean up your data using this manual correction mode. When you are done, then you go back to auto mode, like here, but you will see some of these data are already shown with the little X, so that indicates that here these data were manually corrected, and the corresponding rise times, inter-event intervals, and areas and other parameters are also crossed over. Finally, I can show an interesting feature of the program that can be used to create animations. When you click on the top panel using the right mouse button, then you bring up this pop-up menu, and then you can click here, Animate Trace Sweep. So let's do that. And basically the program will run through the entire experiment, and it will create frames, and these frames will be used to create the GIF animation. So this is like an animated GIF that can be used in a PowerPoint presentation, and it can be really nice and interesting way to demonstrate your data and your findings. You can create animations for the bottom panels too. For example, here you want to animate this plot, the inter-event interval, and then the program will go through the experiment again, and then it will put all these frames into the animation. When this is done, I will show what is the output. These two files were created by the next GIF utility that is shown here. So the animated GIF files are this one, the first, and it shows the sweep, the current trace sweep as we go across the entire recording. So this will show the detected events and the baselines and their peak values as a function of the time. The second one is the inter-event interval as a function of the time. So these animated GIF files can be inserted into a PowerPoint presentation, so you can illustrate your recordings in a convenient and visually attractive manner. I am still developing this program and try to improve it further. I would appreciate if potential users contact me and give me a feedback on how to make this more stable, robust, and more user-friendly. The strategy is basically that I would like to make this analysis as fast and as convenient as possible. On the other hand, it has to be reliable and stable, and definitely I would like to maximize the quality of the analysis. So to do that, I hope that the potential users will send me feedback or questions so I can further improve this program.